Hello everybody and welcome to today's webinar by the Global Antibiotic Research and Development Partnership GARDP. I'm Laura Piddock, GARDP's Scientific Director, and I'm delighted to host and moderate this webinar on biofilms. What are they and why do we care? So Revive is a programme in our scientific affairs team, which is effectively an online space to connect and support the antimicrobial discovery R&D community. And we aim to not only support everybody, but facilitate learning, sharing knowledge and connecting people. And these webinars are part of our educational and outreach activities. All of our webinars are recorded and they're freely available after the event. Please see the website link here if you wish to listen to any of these again in the future. We also do a number of other activities within the team, and these include antimicrobial viewpoints, and there's a few examples shown here. And they're on any topic within antimicrobials, from addressing resistance in cancer patients to what's been going on during the COVID pandemic, strengthening regulation, right the way through to some basic science topics. The link is here again, so please do take a look. We also have a variety of different resources, including the encyclopedia. And this has a table face like this, where you can click on the name of anything, or you can search for a term that you're not familiar with. So you may hear an abbreviation or an acronym such as MIC and not know what it means. If you put it in, it will pull up a definition. And some of these definitions have links to other areas, such as if we're talking about an MIC UCAST or CLIS, and also a video by one of the Revive experts. So moving to our webinar today, you will be able to ask questions live to the presenters immediately afterwards. I do apologise, this has gone too quickly. So if you wish to submit questions, please submit them via the window that says questions on your right. All of the questions will be reviewed, and we will try and respond to as many of them as possible during the Q&A session. If you're particularly addressing a question to one of the speakers, please include their name in your question so that I know who to ask it to. So I'm delighted to welcome Mark Webber and Freya Harrison, who are going to present today. Mark is going to talk first on biofilm adaptation, the good, the bad and the ugly, and then that will be followed by Freya, who will talk about building and using models of lung and wound biofilms. Our first speaker is Mark. He's a group leader at the Quadram Institute in Norwich in the United Kingdom. His research group studies molecular mechanisms of antibiotic resistance, focusing on understanding how, where and when and why bacteria evolve antibiotic resistance. And he has a particular focus on bacterial biofilms and how they respond to antimicrobial pressure within them. He uses a variety of different approaches, from phenotypic to genotypic approaches, conventional microbiology through to molecular microbiology and genomics. And he's looking at the linking between bacterial survival and resistance mechanisms in biofilms. He's published many articles, over 100, and he's acted as an editor for various journals. So welcome, Mark, and we look forward to your presentation now.
Thank you, Laura, for the kind introduction, and for everyone at GARDP for helping put this together. And it's a pleasure to talk today. As Laura said, I'm based at the Quadram Institute in Norwich in the UK. So this is one of the UK government-supported research institutes. We were formerly the Institute of Food Research before changing our kind of mission and moving into this nice new building a couple of years ago. So I'm going to talk today a little bit about what biofilms are and a little bit about why they're antimicrobial resistant and what that means. So a lot of people listening might already know a lot about some of this. So to start with, I'm going to try and give a relatively gentle introduction and then finish with a little bit of data that we haven't published yet. So if you are very genned up on the subject, then there's some new stuff at the end. And if you're not, then hopefully we'll be able to take you through from kind of first principles. So firstly, kind of, well, what are biofilms? So this kind of electron micrograph here shows some, this is salmonella actually, and here you can see that you have these individual cells, but they're not swimming around in liquid in isolation. They are encased in this kind of matrix that they make, and they're stuck together. And actually, this is the kind of very common mode of life for bacteria in the real world, is that many bacteria will form these aggregated communities. And when they do that, they will make a matrix of kind of cement, if you like, of lots of different things that they will produce to encase themselves with it. And this is a biofilm. And biofilms are found pretty much anywhere. So anywhere that you can find life, you'll find bacterial biofilms. So that could be a wet environment, a dry environment, biotic, abiotic. So we find them on animals, in animals, as well as just in environmental surfaces. So as well as just being a fundamentally important mode of life, it actually affects human health and wealth a lot. So these are clinically very important. So infection prevention and control spends a lot of time trying to deal with biofilms. So in a clinical setting, bacteria will survive in a hospital environment very well in biofilms, and often we're trying to break chains of infection by eradicating bacteria in a biofilm. Industrially, they're very important, either in causing a problem from contamination of products or production lines, but also we can use bacteria to make things when they're in a biofilm. So it's a very efficient way sometimes to make drugs and things like that. So these are very important organisms. When bacteria grow in a biofilm, then really the lifestyle is very, very different to what happens when the bacteria, the same cell, is floating around in liquid. So this really is a kind of distinct lifestyle. And all the cells within a biofilm are not the same. Often these are polymicrobial, lots of different species. And even if it is a single species biofilm, the behavior of cells within it can vary a lot. And one of the hallmarks of bacterial biofilms is they're usually very tolerant of antibiotics, very, very hard to kill, which we'll talk about why that will be, because obviously that's something that we're very interested in an antibiotic resistance perspective. Okay, so here are just a few pictures just demonstrating some of the variety of biofilm structures that you can find. And these are all taken from Biofilm Brain Hub, which is a website that the UK National Biofilm Innovation Centre and Quadrum helped to put together. And so these images are all available there. And you can see you've got examples of a couple of environmental biofilms, one from a dry environment here in the top left. This is a surface biofilm where there's not many liquid, and down here on the bottom left you have a stream where you can see the bacteria actually oxidizing and creating these orange structures. You have a couple of clinical biofilms. So this green picture is the inside of a catheter that's been encrusted with biofilm and completely blocked. So this is kind of bacterial biomass inside a catheter. And in the top right, the yellow cells are bacterial isolates in a porcine wound model infection. You see the bacteria covering the wound. And then these last two examples are kind of food spoilage biofilms. So we have examples of biofilms growing on I think this is a butternut squash soup, and on a piece of chicken. So different biofilms will form in different contexts. The structures will be very different, but bacteria are very good at forming biofilms. So how does this happen? How do you go from cells free living to a kind of adhered community? Well, again, it depends a lot on the conditions, the species, but there is a kind of generalized lifestyle that most people will accept, which covers initial colonization of a site and then commitment to changing the expression of genes that are needed to allow you to live in this kind of sessile lifestyle, production of biomass, so more cells and matrix to surround you, and then eventually, finally, a release of cells to allow colonization of new environments. This is a diagram from a review that Paul Stoodley's group put out a couple of years ago, which kind of encapsulates the events that are happening in these different stages quite nicely. So you're kind of from planktonic cells to then initial adhesion, where lots of adhesion molecules are very important, the development of the biofilm by either recruitment of cells or maturation and growth of cells within it, production of biomass to make the mature biofilm, before for cells and aggregates get released to colonize new environments. And understanding this biology is important to being able to come up with ways to intervene, to either inhibit, block, or promote, depending on what you want to do a biofilm. Okay, so I mentioned at the beginning that AMR is a hallmark of biofilm infections, and that they are inherently very antimicrobial resistant, very hard to kill, and the same cell that if you grow it in a planktonic culture might be inhibited quite easily can be orders of magnitude more difficult to kill in terms of the concentration of drug needed when it's in a biofilm context, and that's with no genetic change. Again, this is due to lots of reasons. There's no kind of single universal answer for why this is the case, but some of the common factors that are thought to be important are kind of summarized on this slide, and I'll just spend a minute talking through these and then pick up on a couple of examples. So when people were first studying biofilms, one of the things we realized or was proposed was that actually biofilms, cells in a biofilm are not very easy for the drug to access, so they have kind of low permeability. You see here you have some cells, the antibiotics in red, getting stuck in the matrix, and for some drugs that's important, but for others probably not so much. You have this fact that the physiology of cells is very different when they're in a biofilm context. So here again, this little diagram, you've got a target being produced in planktonic cells and being inhibited by a drug, and actually when they're in the biofilm context, that target or a different version of it's being made, so the physiology is that the cell has changed. Antibiotics often will target a specific protein or enzyme very, very specifically. If they're not making that or it's being changed, then you may become tolerant due to the essentiality of the target being different. Persister cells, so we'll talk more about that in a second, but persister cells represent a subfraction of a population that are in a kind of spore-like state almost, and are very, very, very hard to kill, and biofilms have a high proportion of persisters. Similar but not the same is kind of dormancy, this idea that metabolic activity within a biofilm changes a lot. So you've got this kind of diagram with cells at the edge growing more quickly and kind of green, and ones at the bottom are kind of red and not doing very much. And again, often cells that are metabolically quite asleep and not doing very much are a bit harder to kill. And finally, horizontal gene transfer, so movement of resistance genes amongst members of a biofilm can happen at reasonably high frequency. So you can have cell-to-cell contact being promoted, you've got a kind of plasmid moving between there, you can have cells releasing DNA into the environment, so that's often a structural component of the biofilm. Some species can then take that up, they're naturally competent, and you can have phage moving bits of DNA between members of a biofilm consortium. So all of these are relevant, and the relative importance for any one of these mechanisms will depend on the specific conditions, the bacteria and the drugs, but often it'll be a combination of these factors that together explain why a biofilm is so hard to kill. So just going to pick up on a couple of these in a little bit more detail. So low permeability, this is some data from a paper from 10 years ago now, which showed that actually the matrix production is important, but it depends on the drug and also the strain. So here we have two different Pseudomonas strains, PA14, PA01, forming a biofilm, which is shown here in this flow cell model. Then if you disrupt PEL, which is a polysaccharide part of the matrix in these strains, you can see PA14 quantifying the biofilm, it drops a lot, and the structure is very much changed. These final panels are just when you replace the genes on plasmids to give them back to confirm that this is a direct effect. But if you look at PA01, the effect is not really seen. So PA01 doesn't really suffer from loss of PEL in this context. Interestingly, over on the right, if we look at sensitivity to some drugs, we can see that PA14 is, when you add tobramycin, is knocked back in terms of viable cells. And if you don't have PEL in this white column, then the effect is much bigger. So the drug is much more active in this case, so more drug is able to get to the target, it's the interpretation. Again, the grey bar is just giving the gene back. However, for ciprofloxacin, a different drug, it doesn't make any difference. So it doesn't matter whether you have PEL or not. And interestingly, at the bottom, PA01, the other strain isn't actually affected in this case. So this just shows that the matrix permeability barrier can be important, but it really depends on the bug and the drug. And so just the second mechanism I wanted to talk about in a tiny bit more detail is persister cells. Persister cells are found in most bacterial populations, a kind of small number of here in this diagram from this nice review are shown in red. And this small fraction of cells that are in this persistent state will occur stochastically just in normal growth, but under stress conditions, the fraction will be higher, and in biofilms can be as many as 1% of the total population are in this persistent state. There's a lot of research into how persisters occur. Toxin, antitoxin systems are very important, and the triggers that put bugs into persistence, but also wake them up. And it varies again by species, but the bottom line is that persister cells are very common in biofilms, and they're very, very resistant to antibiotics. So again, hard to kill. So we can see that biofilms are inherently quite resistant to most drugs. So one of the questions that we wanted to ask a couple of years ago now was, well, what happens if you expose biofilms to low levels of antibiotics? So, and this is followed really from work that's been happening in the antibiotic resistance field for the last maybe 10 or 15 years, where it became appreciated that actually low concentrations of antibiotics are actually quite common in the environment, but also can have a very strong selective impact. And a lot of this work was originally done on planktonic populations, so cells in liquid culture, where it's shown that actually exposing bugs to very low concentrations of antibiotics could actually select for resistance quite readily. So bugs do care. But we wanted to know, what about biofilms? So biofilms are, maybe 1,000 times less sensitive to the drug, so maybe they don't care at all. So to address this, we have adopted a biofilm evolution model. So this is a model that Vaughan Cooper in the US first described for looking at evolution of biofilm phenotypes. It's a very simple model where essentially you grow bacteria on a bead and they will form a biofilm. And here you can see you have some green beads. You then will take one of those beads once a biofilm is formed. You'll put it into some fresh media with beads that are marked in a different way, so they're a different color or a different size or shape or whatever. And then you let them form a biofilm, and so on and so on and so forth. So to keep going through the experiment, the biofilms have to form on a bead, but they also then have to colonize to colonize a new bead. So you capture all the bits of the life cycle. It's a very simple model. It's very adaptable. The beads can be made of whatever you want. You can have the media with whatever you want. You can do whatever growth conditions. You can run lots and lots of these in parallel. You can do it in any organism you like. And you can change whatever the stress is. You can have a constant stress, an escalating stress, whatever. And so we use this model quite a lot to understand biofilm evolution. And one of the nice things is that we run these experiments through a period, and then at time points, we will then recover cells from them and we will phenotype them. So we will then kind of put them into a 96-well format. We can then print from that kind of reasonably good throughput for looking for drug sensitivity. So we can print on lots of different plates with different concentrations of drug. We can look for morphology. We can look for staining for biomass. And then we can find interesting things, to sequence and then understand the genetic basis. So this is a model that we've used a lot, and we published this earlier this year, if anyone wants to know more about it. And this is just some information showing from an experiment we did with Salmonella again. Colonies at the beginning of the experiment look kind of as we would expect to start with, and then by the middle and the end point, you can see colonies we recover are making much bigger colony morphologies with much more biomass, and if you measure the amount of biomass that they form, then it gets higher and higher. So the model selects for making more biofilm. Okay, so what did we do? We took Salmonella and we exposed independent lineages, lots of them, to different drugs, to either cefotaxime, azithromycin, or ciprofloxacin, as well as controls with no drug. And we looked to see what happened over time. And so just going to talk you through what this looks like. So here we've got what happened after exposure to azithromycin. And essentially this vertical line means no change. Anything to the right is a kind of log scale of increased MIC, so the minimum inhibitory concentration for the amount. This is the amount of drug needed to stop growth for a panel of different drugs. And so you've got a kind of rainbow of colors. And so essentially each drug is a different color, and the size of the bar relates to how big a change in susceptibility there was. So that the higher the color to the right, then the more tolerant to that drug the bug became. Anything to the left would have got worse, so it become more sensitive, not more resistant or more tolerant rather. And so just a quick look at this first to say that the control data for lineages that were exposed with no drugs is here, but you can't see anything, so nothing changed. Their sensitivity stayed between the early, middle, and late point just the same. So nothing. So just making more biofilm doesn't make you any more resistant to anything. And then we've got planktonic control cultures here at the middle, and these bugs are exposed to azithromycin. You can see that they become more colourful through time, and at the end you get this kind of rainbow, and red is azithromycin, so they become more tolerant to azithromycin, but also other things. And our biofilm lineages look similar, but they actually become tolerant earlier, so they get change to a whole bunch of drugs, and they get this kind of nice rainbow color, so they get kind of multi-drug resistant to a whole bunch of different drugs quite quickly. So that's azithromycin. We did the same thing with cefotaxime, and you can see again nothing happens in the controls. This time you see there's quite a different pattern between bugs that were grown planktonically, so exposed to the drug in liquid, and those that in a biofilm. You see the biofilms have quite a broader pattern of drug tolerance. There's some collateral sensitivity here, so you can see in this case cefotaxime is this kind of orangey brown color. You can see they all become more tolerant to cefotaxime, but the planktonic lineages become particularly more sensitive to azithromycin. And then we also look to see what happens to ciprofloxacin, and this time you see actually a very similar pattern between planktonic and biofilm. So they both become resistant to ciprofloxacin and nalidixic acid, which is in the same class. And there's a little bit of more color in the biofilms, but they're relatively simple. So the first question we had was, well, do biofilms care at all? And these are all exposed to concentrations of the drug that are below the MIC for the wild type. So they do care. Low concentrations of drugs select for resistance in biofilms very quickly, and sometimes the pattern of resistance is different to that you see in planktonic growth. But one of the things that we're interested in is what's the consequence for this? So if you become resistant to something, fine, but does it have other problems to you? Does it affect other phenotypes? So this is over here just exactly the same data I've just shown. So you can see that the MICs, this is resistance to antibiotics, it changes. But we wanted to look to see what else happens to the cell. So now we looked at their biofilm formation. So if you remember the controls here make more biofilm over time. So we know the model selects quite strongly for making more biomass. And if you then look at the evolved populations, the planktonic ones have had no selection for biofilm, so they don't change, which is no surprise. But here, the azithromycin exposed biofilms, they've become tolerant to the drug, but their ability to form biofilm has stayed constrained. They're not been able to make more biomass as the model would select for in the absence of drug. And if you look at cefotaxime, you get something really quite striking. So again, the controls make more and more biofilm, as you'd expect, but both the planktonic and biofilm lineages are awful at forming a biofilm. So they've not just not got any better, they've got worse. So developing the resistance to the drug here has carried a very large cost to biofilm formation. And this has happened, the planktonic cultures, of course, were not in a biofilm context, so they've got no selection to maintain biofilm. But we know the biofilm cultures were under quite strong selection to try and make more biofilm, and they've actually had to deal with the drug, but it's left them much worse at forming biofilms. So there's a kind of collateral sensitivity here. And ciprofloxacin, again, this time the biofilms were able to make more biofilm over time, and not quite as much as you'd normally see in the controls, and the planktonics got worse again. So there's a difference. So the mechanisms of resistance are a little bit different, and the outcomes in terms of fitness are a little bit different. So I'm just going to finalize explaining a little bit about one of the mechanisms. So as I said, we sequenced lots of the outcoming mutants from these experiments to try and understand what had happened, and this tree just shows how they all started from the same ancestor strain where they ended up, and you can just see they're colored by drug, that there's no single universal branch that happens in all the biofilms or anything. It's drug specific. So drugs select for specific pathways of tolerance, and then within that there's some differences between biofilms and planktonics, which are the dark or light colors here. So there was no universal mechanism of resistance, but there were some things that we did see time and time again. So we saw changes in the multi-drug efflux system and regulators of that, and changes in a system that controls membrane permeability. So there are some things that we see repeatedly. So just the example of the azithromycin data. So azithromycin is a macrolide antibiotic. It acts inside the cell, so it needs to get inside to cause its effect by attacking the ribosome, and it's important for salmonella. So what we found in our results, if you remember, you saw a kind of a first step and then a second step selection of resistance, and we found in our first step mutants they all had in different lineages a change within ACRB. So ACRB is a efflux transporter that pumps out antibiotics, and we know can export azithromycin, and they all had a change within this pump, which we interpret to make the pump better able to export the antibiotic. And then what we found for the strains that had a high RMIC and had a range of change to different compounds is they all had this mutation, but now they had another mutation in this regulator RAMR, and this regulator controls expression of the pump, and what you end up with is making more of this pump. So you mutate a pump to be better able to export the drug, and then you produce more of that pump is what we think is happening. And some structural analysis of the change that we find in the pump, which is at this residue here, this R717 residue normally sits in the binding pocket for azithromycin, which is docked in yellow, and kind of constrains export out of the pump and up into the outside world. The mutation that we find results in this L717 substitution. You can see the path out for the drug is now much easier. So energetically, it's easier to get rid of the drug. Okay, so we know that overproducing the RAM system, which is what we see in these mutants, has an impact on lots of things in the cell. So coming back to why do these cells make really bad biofilms? Then we actually know that if you overproduce RAM-A, so this is some data from actually a long time ago when I was in Birmingham, where we can see expression of RAM-A in response to different concentrations of a drug that we know ends up inducing RAM-A expression or ending up with more production of RAM-A. And you can see that that correlates with a kind of downward trend in biofilm formation. And RAM is able to control the expression of lots of genes, and some of the outputs from that are a shutdown and some of the matrix production genes that allow cells to make biofilm. So we understand how this resistance mechanism results in a cost to biofilm. So just a kind of quick introductory whistle-stop tour to biofilms, and biofilms are important and they're everywhere. They are very drug resistant due to various factors, and then some stuff that we've been looking at to show that they really do care about low concentrations of drugs. There is no single mechanism of resistance to antimicrobials in biofilms, and actually understanding this fundamental biology will be important because we will be able to understand how drugs act against biofilms and the fitness outcomes that might be selected by different treatment regimes. So that's all I wanted to say today. Hopefully that's going to prime and set up Freya's talk nicely, and I just wanted to say thank you to this is all the people in my group at the moment, and the work I talked about today is all done by Eleftheria Trampari here in the top left, and then various kind of people who help funding the work in the lab. So thank you very much. That's all I wanted to say for now. So thank you very much.
Thank you, Mark. That was excellent and covered many different areas, which we'll be picking up on in a minute with questions. We're already getting questions coming in. So we'll move now to Freya's presentation. So Freya is a principal investigator in the School of Life Sciences at the University of Warwick in the UK. And she has an interest in how the development of biofilm infection is influenced by interactions between the infecting bacteria, between bacteria and their environment. And her group builds and employs different models of biofilm infections, particularly to explore bacterial physiology and how these can result in highly drug-resistant phenotypes. Her group also uses bespoke models to test antibacterial activity, including natural products. And some of you may remember there was quite a lot of interest a couple of years ago in ancient remedies. And I think Freya, you became a TV star for a short while. So this approach to antimicrobial discoveries is fueled by collaborations that bridge these traditional sciences and the humanities and have led to the work she's doing now. Thank you, Freya. We look forward to your presentation.
Thank you very much, Laura, for that introduction, and thank you, Mark, for a fantastic setup for moving on to this talk. Good afternoon, everybody. I'd like to start the talk by introducing you all briefly to my lab group at Warwick. I'll name check them as we go along and I show their work. And the work I'm going to show you has been funded by UK Research and Innovation, by the charity Diabetes UK, and by the National Biofilms Innovation Centre. So in my lab, we're interested in two particularly important examples of biofilm infection. And if my slides are going to behave and let me move, those are biofilm infections in diabetic foot ulcers and biofilm infections in the lungs of people who have the genetic condition cystic fibrosis. So if you look at the top right image, this is a confocal image of a tissue biopsy from an infected diabetic foot ulcer. And you can see here beautifully the red labeled bacteria embedded in their green extracellular matrix of exopolymers that they've made within the host tissue. And within the cystic fibrosis lung, the small airways, the bronchioles, become plugged with this greenish mass of biofilm and mucus. And if we have a look at the bottom right panel, this really shows the protective nature, I think, of biofilms for bacteria. This image shows a section of explanted CF lung, and you can see Pseudomonas aeruginosa bacteria in red in this big blob of biofilm, which is completely excluding the host immune cells labelled in blue, so the host immune cells can't remove that biofilm. Now, despite the very different anatomical and physiological contexts of these two infections, there's some common themes in biofilm, in CF and diabetic foot infection. Both of these infections occur in sites where the host's normal immune defences and clearance mechanisms are compromised. They both occur in infection sites that are biochemically abnormal. So cystic fibrosis causes the respiratory mucus to become much thicker, stickier, saltier and richer in nutrients than normal, and diabetes causes an infection site with poor vascularization, elevated sugar levels. And the upshot of this is that you can get an infection in either of these areas which lasts for months, years, even decades, despite repeated antibiotic treatment. And it's commonly known that even if isolated bacteria from swabs from these infections are susceptible to antibiotics in standard diagnostic tests, those antibiotics often have no effect when you give them to the infected person. And you see quite a few of the same pathogens in the diabetic foot and in the cystic fibrosis lung. So these are your very flexible, opportunistic pathogens from the escape group, species like Pseudomonas aeruginosa, Staphylococcus aureus, etc. And these infections are often biodiverse. Both CF and diabetic foot infections impose a massive health and economic burden. So I've just picked this photograph from Twitter from a lady called Kate Eveling who has CF, and she's posed here with all of the medications she has to take to stay healthy. And you can see down on her right piles and piles of boxes of antibiotics, and people with CF need repeated doses of oral, nebulized and even IV antibiotics, but those ultimately don't control the infection. So about 70,000 people worldwide live with CF, and sadly 90% of them will die from respiratory failure. The burden of diabetes is even bigger, so you're looking at just under 400 million people have diabetes worldwide. About 10% of them will get an ulcer on their foot or leg that becomes infected. Many of those, that infection will be essentially completely untreatable, completely resilient to any antibiotic. And if that happens and you have a recurrent ulcer, there's a 50% chance of mortality within 5 years because that infection can spread and cause sepsis. And the way to stop that happening is for the person to have an amputation, which is life-altering. So we desperately need new and improved biofilm breaking therapies for both cystic fibrosis and diabetic foot infections. This is why we study them in my lab. So clearly, there's bacteria that are going to find themselves in cystic fibrosis lung or a diabetic foot. The same bacterium colonizing one of those areas could find very, very different infection contexts, very different environments. And these opportunistic pathogens will flexibly adapt their physiology, their gene expression to the unique environment of a diabetic ulcer or the CF lung. So your kind of cartoon biofilm, when it finds itself embedded perhaps deep within tissue, it will find a different environment from when it's living as a blob inside mucus in the very salty, osmotically challenging environment of a cystic fibrosis lung. And so you would expect that the same pathogen might show a very different pattern of gene expression in those two environments, and it might make a different complement of biofilm matrix polymers to surround itself in those two different environments. And all of these things are going to affect antibiotic sensitivity. And I think that's really important to bear in mind when you think of this general problem that when you're trying to develop a candidate new antibacterial compound in the lab, most of these will ultimately fail to translate to clinical usage, especially when biofilms are concerned. So perhaps if we can make more context-specific biofilm models, models that better reflect that environment in a specific infection site, perhaps that will let us find better methods of drug delivery, facilitate drug discovery for drugs that will work in that environment, and help us better understand how antibiotic resistance evolves in those different environments. So my lab uses high validity models of these two infections in our work to try and better understand the fundamental microbiology of common biofilm pathogens and to help us discover novel therapeutics. We developed our own ex vivo cystic fibrosis lung model. And what we do is we take lung tissue from pigs that have been slaughtered for meat. So we get this from a commercial abattoir. It's a waste product from the human food industry. We dissect out small 5 to 10 millimeter sections of the bronchiole from the pig lungs, and we combine that with synthetic cystic fibrosis mucus, which is a very defined medium that mimics the mucus inside the lungs of people with CF. We can grow biofilms of bacteria quite nicely around the lung tissue in this model for days to up to 3 weeks. You can see here an example of a beautifully structured Pseudomonas aeruginosa biofilm with this 3D structure of biofilm layers and channels on top of the epithelium of the lung tissue here. We can do all sorts of downstream assays on this biofilm. We can expose it to antibiotics, then recover the biofilm cells from treated and untreated tissue to enumerate them. We can assay the supernatant of the model for secreted products, virulence factors, etc. And we can image the biofilm in situ. We've published quite widely on how we've validated and used that model if anyone would like more detail. The model of chronic wounds that we use for our work on diabetic foot ulcers was developed by another research group, and this has been published, and it combines synthetic wound fluid, which is a liquid made from fetal bovine serum and peptone water, so it mimics wound exudate, and that's combined with collagen to solidify this wound fluid into a small jelly-like plug, which you can put in a standard microtiter plate, 24-48 ml plate, infect with bacteria, here Pseudomonas, and form a biofilm. And like the lung model, you can grow mature biofilms in this model and then treat them with different agents, add an enzyme to break down the collagen and recover the cells to enumerate and see how many you've killed. So I'm going to use these models to tell you about why we found how using a tailored biofilm model is very important in drug and drug target discovery. And I think it's important to bear in mind that if you use a really good model of the biofilm you're interested in and use it early in your work, you can avoid false positives and you can avoid false negatives. And by that, I mean, often when you use a model that's too simple or too general purpose, you might find lots of candidate drugs that look like they're going to work brilliantly in vitro, but they actually fail when you put them in a more complex in vivo environment. And by false negatives, I mean there might actually be some drugs or formulations that would fail standard in vitro testing, but would actually prove efficacious in vivo in at least some contexts. And if you can model that context in the lab, get the bacteria in the same state as they would be in the host, you will actually find that target. So this is going to be a two-story presentation. Story one is how we've used the ex vivo CF lung model to better understand the physiology and the antibiotic tolerance of Pseudomonas aeruginosa biofilm in CF. And story two is going to be how we've used both the lung model and the wound models in our work on discovering antibacterial natural products. So starting with story 1, Pseudomonas aeruginosa in the cystic fibrosis lung model. I'm going to show you some work that we did in collaboration with Andy Edwards and Akshay Sabnis at Imperial College London. We were looking at the efficacy of colistin for treating P. aeruginosa in the lung. Colistin is a cationic antimicrobial peptide, and it's used very widely as a first-line treatment for P. aeruginosa, chronic colonization in cystic fibrosis. So the first thing we did, we wanted to contrast how Pseudomonas responded to colistin in a simple standard in vitro model versus in our lung model. And so we started off with a Calgary biofilm device, and this is a really simple high throughput way of growing biofilms in the lab. And it uses little plastic pegs that sit in the wells of a 96-well microtiter plate, which you can fill with your synthetic CF mucus, grow biofilm on the pegs, then expose them to antibiotics, see how many die. And then we also tried growing biofilm on our pieces of lung tissue and moving that mature biofilm into colistin and seeing how many bacteria were left alive. So here's data for one of our standard lab strains, PA14, and we found in the lung model, PA14 biofilms could tolerate very high concentrations of colistin, up to 160 micrograms per mil, and that's tenfold higher than the minimum dose of colistin that was required to eliminate them in the peg-lid Calgary device. One example of several cystic fibrosis isolates of CF we also used in these experiments. Again, this isolate could tolerate 10 times higher concentration of colistin than it could in the Calgary biofilm device. So we're getting a much tougher biofilm by using a more in vivo-like model. What we then wanted to ask was, could we separate this increased tolerance of the lung-grown biofilm into reduced penetration into the matrix and increased physiological tolerance to the drug once it got to the cells, as Mark beautifully explained earlier? And this was made possible by Akshay's work, where he made a fluorescently tagged version of the colistin molecule. So we could grow our biofilms around the lung tissue, expose them to the fluorescently tagged colistin, wait some time, then remove the piece of tissue with the biofilm around it, homogenize that, measure how much of the fluorescent signal was in it, and then take also some of the synthetic CF mucus that was around the tissue, measure the fluorescence in that, and work out what percentage of the drug that we'd given had actually entered the biofilm matrix. And we found actually that only a maximum of about 19% of the supplied dose could even enter the biofilm matrix. So this reduced penetration is probably a big factor in why colistin is not very effective in that model. But reduced penetration isn't the full story, because after we published this data, my PhD student Jenny repeated the experiment with one of the biofilm mutants which Mark introduced earlier, which is a Pele mutant that lacks one of the structural polysaccharides for making biofilm. And you can see its dose response as biofilms in the lung model to colistin is identical to the wild type. So we thought, fantastic, we've got this lovely assay for looking at drug penetration that could be really good in helping us discover new adjuvants that aid penetration, help us optimize dosing and find ways to add an adjuvant to colistin so you don't have to give patients so much and it's more effective. But then Jenny beautifully showed if we want to understand more about Pseudomonas biofilm in the lung model, reduced penetration is not the full story. So another PhD student in my lab, Neve, has just finished a really nice transcriptomic experiment where she looked at the expression of nearly 6,000 genes in Pseudomonas aeruginosa over 7 days of infection in the ex vivo lung model to see if she could tease apart what is it doing, exactly what genes, what aspects of its metabolism and physiology is it changing to make it so antibiotic tolerant. And she compared that with the transcriptome just in synthetic CF mucus in vitro. And you can see on this PCA plot that the populations from the lung-grown biofilm in pink had a very different transcriptome from the same bacterium grown in vitro in the synthetic CF mucus. And the thing I thought was really cool about this experiment was that if we look over time, the dynamics of how that transcriptome changes over days of infection in the lung has a completely different trajectory from how the transcriptome changes over time in a test tube. So we're getting an environment where the bacteria are doing something very, very different from what they would do in a test tube. Here's a snapshot of some of the differentially expressed genes in lung biofilm versus in vitro at 48 hours, which Neve has analysed. And you can see she's picked out nearly 1,300 genes that are differentially regulated in that biofilm. Within that, we see a lot of very interesting changes. So there are decreases in cell-to-cell communication, quorum sensing, decreases in the production of phenazines, decreases in type 6 secretion, all things that are very much associated with an acute to chronic switch in Pseudomonas aeruginosa. But Neve was also able to pull out changes in the expression of genes which are associated with tolerance to antibiotics or to antimicrobial peptides. So she was finding changes in the surface of the cell and changes in efflux pumps. And there's a preprint of that story available if you would like to see the full story. But at this point, I'd like to move on to story number 2, which I think will really make this point that we need to use the right biofilm model for drug discovery and use it early in testing if we want to make our efforts at drug discovery as effective as possible. So this story is rooted in the work we have done on historical infection remedies, which is, as Laura mentioned, went viral a few years ago. My group is part of a cross-disciplinary consortium, so people from the sciences and people from the arts and humanities, that aims to data mine historical medical texts to discover new natural products with useful antimicrobial activity. And this is really co-led with Christina Lee at the University of Nottingham and Steve Diggle, who's now at Georgia Tech. The first of these candidates, ancient biotics as we call them, that we've looked at in detail, is this remedy, which is written in Old English in a book that was written about 1,000 years ago in the south of England. And it's a remedy for treating an eye infection, a stye in the eye. Now, styes are caused by biofilm forming in your eyelash follicles, and these infections are often caused by similar pathogens that you find in a whole range of skin and soft tissue infections, like diabetic foot infections. What this remedy tells you to do is to make a mixture of garlic, onion, cow's bile, and wine. So very interesting natural products to a microbiologist. I could tell you the story in lots of detail, but I'm going to start simply by saying this mixture does kill bacteria. So just to give you a summary of some of the things it can do, here's some work that Blessing Nonier did when she was my postdoc. And she looked first at planktonic killing of soft tissue pathogens, and then she looked at killing of them in the synthetic chronic wound model that I showed you briefly earlier. So when Blessing just tested in synthetic wound fluid the ability of this mixture to kill planktonic populations of a whole range of soft tissue pathogens, she found that it could completely take out, completely kill a whole range of unpleasant gram-negative and gram-positive bacteria. So good killing of P. aeruginosa, A. baumannii, Staphylococcus species, for instance. What Blessing then did was that she added the collagen to this wound fluid to make the solid little plugs of soft tissue mimic, and she grew the same pathogens as biofilms in that model for 24 hours. Then she exposed them to our candidate ancient biotic and said which of them could it still kill. And the results were mixed. Some species, like the Staphylococcus species, like A. baumannii and Streptococcus, we could still kill when they were biofilms. Some species, like Stenotrophomonas, Pseudomonas, and Enterobacter, we couldn't kill once we'd made a biofilm in that model. We looked at Staph aureus in more detail, and we tested nearly 100 batches now of this natural product cocktail, and it repeatedly kills Staph aureus. And we were actually able to say from using our chronic wound biofilm mimic in the lab that when we move into animal testing, which we did, it's not worth starting off with Pseudomonas. Let's look at Staphylococcus. So we worked with our colleague, Dr. Kendra Rumbau at Texas Tech, to test this against MRSA in a mouse model of chronic wound infection. And the results were very promising. In fact, they've been so promising that we've started safety testing. We've published on that. And we're now writing up a phase 1 human safety trial that we just completed this year to test if this mixture is safe on human skin, with the aim of developing it into a natural product formulation that could be turned into a treatment or a new wound dressing adjuvant for diabetic foot infections. But the reason I'm introducing this work to you now is that it leads on to two discoveries that really underline this need for using biofilm models and using the appropriate biofilm model so that you don't miss really important information in the early stages of drug discovery. So you saw in the earlier slides that you can get quantitatively different results in antibiotic killing in planktonic culture versus the synthetic wound biofilm. For some species, we can kill planktonic but not biofilm cultures. But our work also threw up a really crucial qualitative difference in the results you get when you test this natural product cocktail in planktonic versus biofilm culture. If you simply look at how this mixture is working in planktonic cultures, you find that the compound from garlic, which is called allicin, is responsible almost entirely for the killing of bacteria in planktonic culture. Now, allicin is not a drug candidate. It's been very well studied. It's very volatile. It has a half-life of a few minutes at physiological temperature. And also garlic extracts are not very safe to use topically. So it's pretty useless as a candidate to move to in vivo testing. But what my PhD student Jess Ferner-Pardo found was that this is not the full story when you look at the activity of this natural product mixture against biofilms. What Jess did was she tested several batches of the natural product iSalve against Staphylococcus aureus in the synthetic wound model. Here's data from 5 independent preparations which Jess made, and they all completely clear a mature biofilm of Staph aureus. What Jess then did was she quantified the amount of allicin present in each of those preparations. She found it varied widely, even though they had the same killing ability. What Jess then did was she made up solutions of purified allicin at concentrations representative of what she found in the batches of natural product preparation and tested whether they could kill Staph aureus biofilms in the wound model, and they couldn't. So allicin at concentrations really at the top end of what we find in this natural product cocktail cannot clear biofilms, but the cocktail can. So you need multiple active molecules in this remedy. It's a multi-partite remedy. It really is a cocktail of active ingredients. And we would not have known this if we'd only worked in planktonic culture. We would have stopped very early on because we would have thought, oh dear, we've rediscovered allicin. It's not very useful. We wouldn't have realized that there's some fascinating candidate natural product antimicrobials in this mixture. And the last thing I'd like to show you really comes back to the idea I mentioned right at the beginning, that different biofilm infection environments can trigger the same bacterium to have very different antimicrobial susceptibility profiles. So let's look at Pseudomonas aeruginosa, which is a very flexible opportunistic pathogen. It causes lung infections in cystic fibrosis. It causes wound infections in diabetic foot ulcers. If you're immunocompromised, there's really no organ or tissue in your body which this bug cannot invade. Pseudomonas aeruginosa was one of the species which Blessing found she could kill with the natural product ancient biotic when it was growing planktonically, but she could not kill at all with the natural product ancient biotic when she let it form a mature biofilm in the wound model in the lab. However, we then thought, we've also got this lung model. What happens if we grow Pseudomonas aeruginosa in the cystic fibrosis lung model and then try and treat it with our natural product ancient biotic? And in this case, from a mature biofilm of Pseudomonas aeruginosa growing in the CF lung environment, we suddenly can completely kill this population with our mixture of natural products. So the same bacterium is completely resistant, completely tolerant to this natural product cocktail in one infection context, but very, very sensitive to it in another. So just to sum up, I think as microbiologists, we can agree that there are certain big priorities in in antibacterial research and development. We know that chronic biofilm infections have extensive and quite unpredictable antibiotic resistance and tolerance profiles, and we need better diagnostic assays and better R&D platforms to find agents to treat these infections. And we also need novel agents to treat biofilm infection. We need to reopen that pipeline. So in my lab at Warwick, the ways we're trying to address these priorities really come down to developing and using these very high validity ex vivo and in vitro models of biofilm infection, trying to really build the appropriate physical and chemical environment that pathogens find in a particular infection context. And we think that this will really aid us in discovering new drugs or new adjuvants that could do things like help drugs penetrate biofilms. And something I haven't talked about today, but I'd just like to mention that we'd also like to use these models to look at how resistance evolves to the same drug in different infection contexts. So thank you very much for listening. And I'd be very happy to take any questions.
Thank you very much, Freya. And back again to Mark as well. Thank you for your thorough presentation. So we will now start the Q&A session. A quick reminder to the audience that you have this panel on your right-hand side. This is an interactive Q&A where you can submit your questions in this panel, and then they will be shown to me and I will just ask them in the order that they come. And so just a reminder again, if you want to address a question to Freya or Mark, please put their names first. If it's an open question that either could answer, I will just raise the question in that way. So we're going to start with the first question. I'm going to go to Mark first, just give Freya a little break from talking for a while. So Mark, where selection for drug resistance in a biofilm has a fitness cost, is that stable, or can compensatory mutations occur over time that allow both the drug resistance to be stable and for fitness to be restored?
Okay, thanks. That's a very interesting question. So we did look at that actually for some of our populations which were resistant but were very poor at forming a biofilm. So we kind of did some just biofilm adaptation alone with no drug for those populations to see what would happen. And generally the answer is the resistance is maintained, and they do manage to get back to making more biofilm. And they do that by just breaking some of the regulatory circuits that have been altered. So it is possible for them to develop resistance to drug and become good biofilm formers. So I think probably what that tells us is that if you're understanding the trajectory of this selection and the fitness impacts from it, that you want to make sure that your treatment is kind of short and sharp enough to clear the infection down to a point that it's gone before there's an extended passage window for the bacteria to grow and grow and grow, because they will be able to get back as ever the bacteria seem to find a way.
Thank you. I'm going to ask you another question now, Mark. And this has come from the audience again. So have you also experienced the impact of chemicals other than antibiotics on resistance or tolerance in biofilms? And for instance, biofilms are often within drinking water systems and are considered chlorine tolerant, for example.
Yeah, okay, so yeah, we have. So we've done a lot of experiments with all sorts of non-antibiotic antimicrobials. In fact, actually a PhD student of mine, Gregory Wickham, has done parallel evolution experiments of the type I talked about for kind of 50 separate chemicals, and then seen the outcomes for all of them. And the short answer is there's a very wide spectrum between some things. So some of the antimicrobial metals are actually very hard for bugs to deal with, and they act pretty well. Some other things they deal with very easily. So some of the quaternary ammonium compounds, for example, they'll react and they'll develop resistance to quite high levels quite easily. And there's a huge spectrum within that of collateral resistance and sensitivity to other agents. So some drugs really select cross resistance, some don't. Some have big fitness impacts, some don't. So it really varies. So that work will hopefully see the light of day in the next year or so. But there's but yeah, it's all sorts of things can select for all sorts of outcomes in biofilms.
So there seems to be no hard and fast rule then, Mark?
Not really, and even within a class, so you can kind of sometimes you can say, okay, this group of compounds seem to look similar and this set looks similar, but even so, there are exceptions to some of those rules. I mean, some of the antimicrobial metals are pretty good generally in that context. They seem to struggle to adapt to those, but it varies amongst classes. So if you look at a series of different quaternary compounds, you'll see a couple of them are very good at selecting resistance, a couple of them it's really hard for bugs to do it. So it's quite hard to be very general.
Thank you. So Freya, for you, you've described two models of biofilms, the lung model and the wound model. What are the limitations of these models that you've shared with us?
I think that's a really important question. And one thing I would say about the models we use is that we don't have a host response. So we don't have a live immune system remaining in the pig lung tissue, and we don't have an immune system component at all in the synthetic wound model. So if people want to use those models to integrate that aspect, that needs to be added. What we're really focusing on with these models is getting the right biofilm structure and cueing the right physiological state of the bacteria, but we don't have a full host response.
Thank you. Another question for you is, are these models useful for mixed biofilms?
Yes, they are. So other groups have published quite extensively on mixed species biofilms in the wound model, including some beautiful imaging that shows what a similar structure you get of communities to biopsy tissue from a chronic wound from a person. With the lung model, we've just published a paper showing that we can get a 3-species biofilm of P. aeruginosa, Staph aureus, and Candida albicans in that model as well. And we're also focusing quite a lot in my lab at the moment of looking at mixed Pseudomonas burkholderia biofilms in the lung. So as anyone who's worked with mixed biofilms will know, it is quite tricky, but it can be done.
Thank you. So either of you can answer the next question. What are the common molecular imaging techniques to monitor biofilm composition or even biofilm maturation over time? I mean, you're both welcome to answer it. Who would like to start first?
Oh, okay. Yeah, right. Go on, Freya. You answer.
I was going to say, actually, there's much more of a reliance on confocal microscopy in a lab setting and on standard histological staining and light microscopy in a diagnostic setting rather than molecular imaging. So people are starting to use 3D orbisims, for instance, to do mass spec-based imaging of biofilms, and that's throwing them into really beautifully defined relief. But I think that's an area that we need to focus much more on in the future if we really want to learn about what is where in the matrix, and particularly to look at the architecture of the matrix in different contexts.
Yeah, I should probably give a quick plug to Cynthia Whitchurch, who's a colleague of mine at Quadrum, who's done a lot of work at imaging biofilms with kind of ultra high resolution microscopy, and as a research tool, that's amazingly powerful. For example, to show the role of DNA release in pseudomonas biofilms, for example, and there are other kind of specialist platforms. I think it's Dundee, where there's a kind of mesolens setup that was used to find channels in E. coli biofilms that hadn't been appreciated in the way that they're formed and stuff. So kind of for research tools, there's all sorts of very high level work that you can do with microscopy, but diagnostically, I think Freya's right. It's still kind of relatively simplistic.
So really picking up on that theme, there's another question from the audience is for Freya. Do you consider that we will find a standard model to study biofilms in vitro that could be used directly in clinical microbiology labs, such as those in hospitals? In the same way as automatic methods are starting to be used, and certainly are already used in some countries, to analyze drug susceptibility, will we get to a point where these models can be transferred for routine use?
So if you're thinking of a one-size-fits-all biofilm model, I would say no. I think people have looked quite extensively at whether using the Calgary device, the peg-lid biofilm, combined with host mimicking media actually gives good diagnostic predictions, and it doesn't. So that's definitely been done for cystic fibrosis. And using that model doesn't give you any better predictive power than a standard microdilution, which any CF clinician will tell you is pretty useless. I think where you have good one-size-fits-all predictive models, and that goes for genomic prediction as well, is where you've got acute infections. But I think once you've got a chronic infection that's very adapted to the host and you've formed a biofilm and is very entrenched in that unusual environment, I do think that's where you need the very tailored, specific model for that infection. But that's
sorry, Freya, carry on.
I was going to say there's no reason why some of these models can't be moved into a diagnostic setting. So our pig lung model for CF is already being used by a microbiological services company for pharmaceutical clients in high throughput testing of candidate drugs. So I think if you think about the cost benefit ratio of getting prescribing right for these biofilm infection contexts, I think it does become efficient to use these specialized models.
I'd agree. I think that Freya's quite right that probably for chronic infections, that's where there's value. I mean, the very powerful slide she had of the young lady with the hundreds of products that she's taking for years and years and years and years. There's actually you've got time there to in a kind of personalized medicine approach to understand what's happening in that context. For example, but an acute infection, it's not really going to be timely enough, I don't think, and there's probably is no one size fits all. I would agree totally with Freya.
Thank you. Mark, what potential is there for specific antibiofilm inhibitors and how might they be used clinically?
Yeah, that's a good question. I think some of the data that Freya showed actually is really powerful in this respect, in that actually often you're going to need more than one thing together to have an outcome and an impact. So if you understand the biology of how you can allow access to a drug, or how you can change the behavior of bacteria in the biofilm, that then you can understand how better to kill them. So combination therapy is something I think that is potentially very, very powerful. Historically, people have been a bit sniffy about putting things together, but actually we use combination therapy all the time. So there are many regimes that are specifically drug plus adjuvant, but then also we use many drugs together in seriously ill patients. So I think there needs to be a little bit of a change in just not being so inherently worried about combination therapy. And we don't really understand a lot about the biology of what's happening within cells within a biofilm. So we know quite a lot about where the biofilms are and how hard they are to kill or not. And we really need to understand more about, well, what are they doing to allow them to survive? Can we, so for example, people are quite interested in therapies to try and wake persister cells up. Can we kind of release the brakes that are holding cells in a persistent state and then allow them to be killed, for example? That's one thing that might work. Again, probably won't be a one size fits all for that, because the mechanisms of persistence are different in different species and different cells. But that would be one example where you could see, actually, if you know you can kill 99% of the population, it's that recalcitrant 1%. If you can help wake those up with a drug that is then in combination with a standard antibiotic that now is effective, then you've got a half a chance of clearing that infection.
So the next question sort of does pick up on that, really. Perhaps this is for you, Freya. Do the regulatory authorities, it doesn't state here which country, but I guess for argument's sake, we could say FDA or EMA, have biofilm testing standards for new drug candidates?
Yes, there are certainly in the UK accreditation standards for the biofilm platforms you use and the protocols you use. So those would be administered by UKAS in the UK, and so those biofilm platforms would have an ISO number associated with them. In terms of a benchmark for efficacy of a drug against a biofilm, yes, there are some of those as well. So I know, again, my view is quite UK-centric, but for an advanced wound dressing with an antibacterial component, for instance, that has to show a sustained 4 log reduction in viable bacteria over a 7-day period. for instance. So yes, there are some general rules there.
Thank you. So Mark, this is quite a complicated question that we've had put forward. So I hope I don't mangle it and misunderstand it, but here we go. So you talked about tolerance being an important strategy for biofilms to evade antimicrobial action. Could this strategy be perceived as a fitness effect in terms of collateral sensitivity? So the question is, are they persisters where they're metabolically or replication active? So it's trying to wrap up persistence, tolerance, biofilms, and metabolic sort of dormants.
So you get the spectrum of all of them within a biofilm. You get cells that are persistent, asleep. You get cells that are kind of not doing very much and they're kind of tolerant, they'll survive transient exposure. So I think probably maybe what the question was getting at was if you kill all of the actively growing cells that are probably causing the pathology, then actually if the side effect is everybody is forced into a persistent or tolerant state, is that actually a side effect that means that they're not in, they're less of a problem, right? So that's one possibility. And actually so we do some work talking with the orthopedic surgeons and prosthetic people who have prosthetic joint infections, and there are patients that have a low level rumbling biofilm that they don't worry about. So they're not really actively unwell, and the getting the implant out would cause more trouble than just leaving it alone. It's not affecting their daily life. So there are potentially cases where actually bugs that are sitting dormantly not doing anything you can live with, but the flip side of that is that you retain a reservoir of cells that will wake back up and expand again potentially, and if they start shedding into the blood, for example, it's a source of bacteremia. So I think that was probably the thrust of what the question was asking. So if the selection is for tolerance or dormancy, then okay, that's one thing. But if you have to keep antibiotic therapy there all the time to keep them constrained, painted into the corner, then at some point they will stop working. They'll become resistant to it, or break, or you end up with extremely long-term drug use forever and ever and ever and ever. And I suppose in some ways that's analogous to what happens in CF. It's not really the case that everything's dormant or tolerant, but you use a lot of drugs to hold stuff at bay. But it's, as kind of Freya eloquently said it, you're delaying the bad effect, but you're not guaranteeing it's not going to happen. So I think we should try and aim to do better, if that makes sense. But yeah, it's a sensible question.
Thank you. So this question is not directed to either of you in particular, so maybe you'd both like to pick up on it, but to you, Freya, first. Is there any correlation between biofilm formation, virulence, and resistance observed during in vivo and in vitro studies? So this very much picks up on what you were saying, Freya, that they behave in different ways depending on the context. But it's the virulence part here that's been added into this question.
So I'd say on a sort of grand scale, you do tend to see a trade-off between biofilm formation and overall virulence, so that the bacteria will switch from either a high virulence state to a low virulence and making a biofilm state. But within that population, are there correlations between those phenotypes? I couldn't give you a complete answer to that. I know from one study I was involved in looking at Pseudomonas isolates from a person with CF from one sputum sample, we found that there were all sorts of trade-offs between different phenotypes, and you didn't have one clear correlation between overall virulence and biofilm formation. What you did have was almost specialization within that population of producing a particular virulence factor or a different one.
Thank you. Mark?
Yeah, so we looked at that to some degree with the isolates that we selected in our model, and we used a Galeria virulence model. So this is a kind of moth larvae model that is a kind of reasonably easy virulence model and reasonably predictive. And essentially what we found is that the bugs that had selected to make loads of biofilm, or mutants that made almost none, were both quite bad in terms of virulence. And so if you kind of knock them off their normal range of life. So the wild type will kill the larvae at a certain point. And if the bugs that were adapted, so they had no change in their drug sensitivity, they made loads of biofilm, they weren't very virulent actually. And then the ones that had selected kind of high levels of resistance but were very poor at forming a biofilm as well had a bit of a big, but then that's probably drug specific costs as well. So generally more, I would agree with Freya's kind of generalized statement at the beginning that more biofilm is a kind of, making loads of biofilm is a kind of low virulence state. But again, it's probably difficult to predict too broadly. It would be species specific and mechanism of why they're making more biofilm or why they're resistant to this and that. But yeah, more biofilm doesn't mean more virulence. As again, I think some people have, historically was a kind of idea that kind of, oh, these things that make loads of biofilm, they'll get in, they'll be impossible to get out of the body, etc. But actually, they're in a different state of life. They're sticking, so they're not necessarily highly virulent in that state. But a big population of the bugs will then have lots of changes within it.
Thank you. So there's another question for the two of you. So you both discussed sort of when we were talking about conventional antibiotics, and they tend to work either by killing bacteria or by just inhibiting their growth. But what do you think about approaches that disrupt the biofilm or make the bacteria stay planktonic? You know, is there any merit in that approach? Are there any examples of that approach? Freya, would you like to pick up on that first?
Sure, I think yes, there are. So the first thing that comes to mind would be enzymes that degrade the biofilm matrix. So cocktails of glycoside hydrolases that nibble away at those structural polymers. So there are preparations of those. So Dispersin B, which is already being used as a topical wound spray, is one of those biofilm disruptors. And I think I would say, yes, there is a lot of potential. And when we look in the lab, we do find those kind of enzymes do really potentiate quite a lot of antibiotics that can't normally penetrate the matrix. I don't know if that's the kind of answer you were looking for.
I'm not looking for any particular answer. I mean, we just get lots of questions and we do our best to answer them. So thank you. I don't know, Mark, if you would like to add to that.
Yeah, I mean, people have looked at DNAs as well, which is DNA is often a structural component of biofilm matrices, and there's been some really interesting studies that are showing that's had quite a big impact. And I think generally knocking the biofilm matrix down can sometimes have a really good impact if you've got an immune system that's working with you to try and clear an infection. So there's good evidence in that respect. So I think there's definitely potential for that. And I think the second part was, could you fix them in a non-biofilm state? And possibly. I mean, these are regulatory networks that are well evolved, and it's a big lifestyle switch into forming a biofilm. So that there have been people looking at inhibiting quorum sensing, etc., to try and do. I'm not sure I've seen anything which has kind of managed to fix a bacteria in a non-viable state by a chemical inhibitor, but or that's been applied in any kind of relevant model. You can do it in vitro.
Okay, thank you. So Freya, you showed a series of different experiments with the same process, but with a different bacterial species in it. And you showed that, for instance, Pseudomonas strain 14 in a biofilm in your wound was resistant to the I-salve, but compared to it in a different model was more susceptible. Does this give you any indication of the mode of action, or is it a structural issue?
At the moment, I couldn't answer that. I suspect there's components of both, because the structure of that biofilm and the depth of it, and also the structure of the tissue around it, or the environment around it, is going to vary. But I strongly suspect there's a change in target expression as well.
Thank you. So Mark, you've talked a few times now about cells in biofilms being dormant. Do you think you could give some clarity on the difference between a dormant cell versus a persistent cell?
Okay, right. Yeah, so persistent cells have initiated a kind of shutdown program, if you like. So the cell is arrested, ribosomes have been bundled up. This is kind of toxin antitoxin systems. There's various mechanisms that are really shutting down the cell's machinery. And dormant cells, in my mind, are ones that are just not doing very much. They're kind of sitting. They are not really, they're metabolically not turning over very fast, they're not growing very quickly, but they're metabolically awake, if you like. So, you know, all of the enzymes that are present in the cell, all the cell machinery will be functional, but they're not. So none of it's been arrested by initiating a kind of persistence mode of life, if you like. So persistence is much more dramatic. I think where I'm talking about dormancy, it's much more cells that are really not attempting to grow very fast, they're not turning over very fast, they're probably somewhat starved, they're struggling for nutrients, they're not in a kind of fast growing mode of life, so which can affect the antibiotics' ability to kill cells. So my really rubbish analogy is you know, imagine kind of an antibiotic's like sticking a spanner in the works of a motor, and as the motor's turning, it's very efficient, causes lots of damage. As the motor's not doing very much, it doesn't have so much of an impact.
Thank you. So, Freya, your ancient antibiotic is active against biofilms such as those that cause eye infections or foot ulcers. So, your work is your work going towards a topical agent development?
Yeah, we very much hope so. So we think that's a good thing to aim for, at least in the first instance, partly because the testing and regulations around developing a topical device are a little bit easier. So we're thinking something that could be incorporated into an advanced wound dressing.
Thank you. There's a related question to this, because you showed differential activity in the different models that you used. Can you clarify whether you were indicating that, that using the different models could give you a way of discriminating what the potential indication for a natural product or even a novel chemical entity could be chemically synthesized, could ultimately be and developed to treat?
Absolutely, and I think if you have a range of these models at your disposal, which are cheap enough and high throughput enough to test early on, you can really focus on the most promising route right from the start.
Thank you. Mark, your work that you were showing today was for Salmonella. What parts of your work could translate to other species?
Okay, so we've done the same thing with Salmonella, Pseudomonas, Enterococcus, Staphylococci, and other groups have done it with many other species, Burkholderia. It's quite, the reason I mean, we're interested in mechanisms of resistance, really. That's what we do, and we're interested in trying to understand how can biofilms adapt to this stress or that, and if so, how would they do it? And then you get a starting point to say, well, actually, does any of that matter? So any of these changes, are there ones you see anywhere? So for example, that ACRB change that we saw is seen in patient isolates who've been exposed to azithromycin. So okay, it kind of probably relates to something that actually happens in reality. But the model's very flexible, so you could change, you can, anything that if you can grow it, you can evolve it, if you like. So, and then you can modify the conditions and the timescales and the stresses and whatever. So yeah, we've run it, or we do run it in various organisms, but yeah, it's pretty flexible.
Thank you. So this is quite a long question, so bear with me, Freya, while I read it out. As we know, the resistance of microbes to antibiotics is increasing day by day. It has been seen that traditional medicines have been explored from the last few decades as antimicrobials because of the presence of secondary metabolites. This person wants to know about the use of secondary metabolites and whether this will be a good alternative approach for biofilm inhibition.
So I think the question is probably thinking about plant secondary metabolites and plants that were used in traditional medicines. Yeah, and I think it is definitely worth looking down that route. And I think the big thing is coming back to that idea of combinations and cocktails, because a lot of these plant secondary metabolites, when you purify them, you lose the activity of the original raw extract. And so I know there's a lot of work among ethnopharmacologists about specifically developing assays to look for synergy and to find synergistic combinations of these metabolites.
Thank you. So a question for either of you, but perhaps Mark. What is the role of quorum sensing in biofilm formation and antibiotic resistance? And the second part of that question is it species specific or a general phenomenon?
Yeah, okay, so again, potentially massive, and I'd say it's a reasonably general phenomenon. Actually, Salmonella is an unusual species, and as I talked about Salmonella earlier, it doesn't do well. We don't understand quorum sensing in Salmonella very well, shall I say. I won't say it doesn't quorum sense. It senses and doesn't maybe send that many signals, but we don't maybe understand how it does. But for other organisms, so Pseudomonas, for example, is well understood to be very important in biofilm, can control lots of the resistance mechanisms as well as the behavior and initiation and formation of the biofilm. So quorum sensing is very important. I think we generally would accept that that's important, and there has been quite a lot of work looking to see, can you basically hijack the signaling, can you block the signals, can you destroy them, can that be useful? And the answer is it could well be. So yeah, I would say as a general rule, quorum sensing is quite important, and I happen to give an example from a species where it's probably the least important.
Freya, would you like to add anything about quorum sensing and resistance?
I think Mark's nicely covered it all there, to be honest.
Okay, I didn't know if you wanted to say anything specific for Pseudomonas.
I guess I could talk about Pseudomonas quorum sensing all day, because it seems like every few weeks another part of the quorum sensing system is discovered. So I think with Pseudomonas, I'd say it's got at least 3 quorum sensing systems, and something we don't fully know yet is how the balance of how it's using those 3 systems might change in different situations as you move from an acute to a chronic infection. And that's likely to be important to consider. If you want to target quorum sensing, which system are you going to target?
Okay, well, thank you very much. I noticed that time has really marched on with us, and we are going to have to bring this webinar to a conclusion. Thank you both for excellent presentations, very clear, very thorough, and for answering so many diverse questions.
It's a pleasure.
Thank you.
So, thank you. So we do have another webinar organised for a few weeks' time for World Antibiotic Awareness Week. We will be confirming the date within the next few days. Do hope that everyone who's joined us today will join us for that as well. So thank you again for joining us. Goodbye.